Hello students, welcome to Shri classes. We will begin lecture number 2 and heredity and evolution. In this lecture, let us have a look at Mendels dihybrid experiment. Dihybrid experiment, as the name suggests, it is di means two contrasting visible characteristics or characters. Hybridization means it is crossing. So in the dihybrid experiment, Mendel carried out such an experiment where it involved plants having two pairs of visible contrasting characters, two pairs basically. What is that we learnt in monohybrid experiment? Only one pair of contrasting characters were studied. And here in the dihybrid experiment, Mendel studies the plants having two pairs of visible but contrasting characteristics or characters. For example, height and shape of the seeds were crossed to study the independent inheritance of traits. So, height and shape of the plants were considered here. Shape refers to shape of the seeds basically. Height refers to height of the plant. So, this particular experiment, dihybrid experiment, is to demonstrate the independent inheritance of traits. That should be kept in mind. Whereas, to repeat again, the monohybrid experiment was to demonstrate the dominance of traits or expression of traits. Whereas, dihybrid experiment is to study the independent inheritance of traits. Before we look at this diagram and the Punnett square, what is that Mendel did? Let us study this one. Mendel selected pea plants of two pairs of contrasting characters, namely tall plant with round seeds and short plants with wrinkled seeds. With respect to height, tall plant and short plants is one pair of trait, and with respect to the shape of the seed, round seed and wrinkled seed is another trait. So here we are comparing or we are studying two pairs of contrasting visible characters. So when the pea plants with tall plants with round seeds and short plants with wrinkled seeds, these were crossed in the first progeny, that is first generation, in F1 progeny, he observed that all the plants were tall with round seeds. No short and wrinkled plants were observed. All the plants in F1 progeny were tall plants with round seeds. Thus, can we say that tallness and roundness were dominant traits in F1 progeny? Yes, absolutely. Next, then he allowed the self-pollination of F1 generation plants to get F2 generation plants. Surprisingly, he observed that plants produced in the F2 generation were the combinations of short, tall, round and wrinkled, meaning combination of all these independent four traits were observed in the second progeny in the ratio of 9:3:3:1. So what is that 9, 3, 3 and 1 represent? So which visible traits they represent? We will come to know only after drawing the Punnett square. So later in the Punnett square, we will try to see what is this ratio, phenotypic ratio 9:3:3:1. We will see that later. But he observed that when F1 progeny plants were self-pollinated, in F2 generation, he observed the combination of traits in the ratio of 9:3:3:1. That means all these four traits were independently inherited in second generation plants. Thus he concluded that the traits tallness, roundness, shortness and wrinkled, all these were inherited independently in F2 progeny. So now let us study in terms of crossing of the plants and preparation of the Punnett square. Here the pea plants that were considered were tall with round seeds and short with wrinkled seeds. Let us represent the tall as capital T, and round seeds as capital R. This is with respect to the parents. Short and wrinkled. The contrasting character for tallness, capital T, is small t, which is shortness. And if we represent capital R for round, then wrinkled seed should be represented by small r. So small t, small r is the representation for short and wrinkled plant, and tall and round seeded plants were represented by capital T, capital R. What about the gametes of these two plants? Gametes will contain half the number of genes, basically half the number of alleles also, you can say. So it should be a combination of both tallness and roundness. So the gametes will have only one T and one R, both are capital. Here one T and one R, both are small. So these are the gametes. When these two were cross pollinated, the resulting F1 progeny plants were having a genetic composition capital T, capital R, small t, small r. This is nothing but the combination of these two. So originally in the plants there were 4 genes, and the new generation plants also there are 4 genes or alleles, you can say. So original number of alleles of each plant were restored back in terms of the numbers I am talking about. So 4 alleles were there, so all those alleles got restored by combination of gametes here. So this is the F1 progeny plant's genetic composition. And we saw here in F1 progeny, he observed that all plants were tall with round seeds. So we can say capital T and capital R are the dominant traits, small t and small r are the recessive traits, meaning the tallness and roundness got expressed in F1 progeny, the shortness and wrinkledness got suppressed in the F1 progeny. Now next he performed self-pollination of a plants of F1 generation having a genetic composition capital T, capital R, small t, small r. Here there are 2 plants, tall round. Self-pollination of F1 plants is nothing but the genetic composition is this and this. These are the 2 plants, and the gametes will be, check here, the gametes must contain half the number of genes. Here there are 4, so the gametes should have only half the numbers, means only 2 genes should be there or 2 alleles should be there. In that case it should be a combination of both height and shape. Therefore capital T, capital R represents one gamete, capital T, small r represents second gamete, small t, capital R represents third gamete, and small t, small r represents the fourth gamete. So for this plant, these are the genetic different combinations of the gametes. Similarly for the other plant, these are the genetic combinations, both are same here. Now let us consider one tall round plant over here side and another tall round plant this side. These are the gametes 1, 2, 3, 4, 1, 2, 3, 4. Similarly 1, 2, 3, 4. These are the gametes here. Now you combine this and this in these blocks. Capital TR, capital TR, capital TR, capital T, small r here, capital TR over here, small t r over here, capital T r here, small t r here. So this is how we can prepare the Punnett square using these different combinations. Now look at, I have already clubbed together certain combinations, I have grouped together certain combinations. Wherever we find capital T and capital R, we know that these are dominant traits. So wherever out of this entire Punnett square you find capital T and capital R, then definitely the plant is tall and with round seeds. So if we count here all these red letters 1, 2, 3, 4, 5, 6, 7, 8 and 9, there are 9 plants with capital T, and capital R combination. See, one capital T and one capital R is enough to make the plant tall and round. Let other two be any combination, we need not worry at all. If one capital T and one R is there, then it is a tall and round. So by counting, we observed that tall and round plants were 9. Therefore, here in F2 generation shows all 4 combinations of traits. The phenotypic ratio is 9. What is this 9 here? 9 represents tall and round based on these red letters which I counted. So tall round plants were 9. Next you look at this blue here. Capital T small r, capital T small r. One capital T is enough to make the plant tall. Any other capital letters in this one, and in this one? No. Capital T is there, capital T is there, capital T is there. So in all these 3 cases, tall plant. Small r is there, wrinkled. Small r is there, wrinkled. Small r is there, wrinkled. Capital T, tall. Small r, wrinkled. Tall, wrinkled. Therefore, tall, wrinkled, 3 plants. Next comes small t capital R, small t capital R. We are having capital R here, capital R here, capital R here. Meaning in all these 3 cases, capital R is the dominant trait for roundness and therefore round seeds. Is there a capital T? No. Is there a small t? Yes, here small t everywhere. Meaning definitely it must be a small short tree. So short with round seeds, 1, 2, 3. Short with round seeds, 3 trees. And finally we are left with all small letters TR, TR, which is short and wrinkled. And therefore the phenotypic ratio is 9:3:3:1. 9 is tall round seeds and 3 plants is tall wrinkled. Next 3 plants short round and 1 plant short wrinkled. So definitely we can see in this case that the tallness and shortness, roundness and wrinkledness, all these got independently inherited. See, initially we took tall and round and short and wrinkled. Can you expect short with round and tall with wrinkled? Yes, in the second progeny we expected tall with wrinkled, 3 plants. Short with round seeds, last one, here is short with round 3, and finally tall with round and short with wrinkled with the genetic combination of capital TR and small tr, where only 9:1 at the end. So what I mean to say is basically here, irrespective of whether the tallness and capital roundness belonging to one particular plant or shortness or wrinkledness belonging to one particular plant, in the final F2 progeny we observed the entire combination of all these things. Tall with wrinkled, tall with round, short with wrinkled and short with round. So finally the phenotypic, why it is happening this? So the phenotypic ratio is basically 9:3:3:1. Tall round is to tall wrinkled is to short round is to short wrinkled. See here, remember dear students, in order to write this 9:3:3:1, I think something is wrong, but let us bear this with this thing. I think I'll check up later. 9:3:3:1, in order to get this ratio, you have to have the Punnett square drawn. I think I'll stop it and now explain. Here it is very important, students, that in order to write this phenotypic ratio, phenotypic means the ratio of observable traits basically. So the phenotypic ratio 9:3:3:1, this combination, when you write this tall round, tall wrinkled, short round and short wrinkled, writing of these should be read from this chart. So unless you prepare this chart, you will not come to know what is that 9:3:3:1 ratio. So finally here we obtained tall round is to tall wrinkled is to short round is to short wrinkled as 9:3:3:1 ratio. That means The whole idea of this experiment is that finally, irrespective of which plant we considered in the F2 generation, in F2 progeny, these tallness, roundness, shortness and wrinkledness were independently inherited and we could obtain this combination. So this experiment is to demonstrate the independent inheritance of traits. Okay, so make a note of this one. I hope you have noted down. Next, one more experiment we will take it. It's a dihybrid experiment. Plants having two pairs of visible contrasting characters, shape and color of the seeds is taken. What is the shape here? Round and wrinkled. What is the color of the seeds? Green and yellow. So we have taken here the combination of the shape of the seeds as well as color of the seeds, and this is the example from your textbook. So let us try to understand this here. These are the parent plants also, or truly bred plants you can say. Round, wrinkled, green, yellow. Please remember, always the dominant traits are represented by capital letters and recessive traits are represented by small letters. If you look at this one, roundness we are using capital R, whereas for greenness we are representing small y here. In this case, the wrinkledness is represented by small r, whereas yellow color is represented by capital Y. Meaning, green color is a dominant, round shape is a dominant shape and yellow color is a dominant color. Whereas capital Y, once again capital Y and capital Y, yellow color is dominant color, and roundness, capital R is the dominant shape. Whereas small y, green is recessive color, and wrinkled, small r is recessive color. What are the gametes of this one? Gametes should have the combination of the shape and color. So capital R, small y, this is one gamete here, and for this small r, capital Y is one more gamete here for this. If you combine these together, we get capital R, small y, capital Y. Only capital R and capital Y is enough to make the plant with round seeds and yellow seeds, and therefore when F1 progeny we obtain all the plants with round shape and yellow color with a genetic combination capital R, small r, capital Y, small y. Now let us do the cross pollination of these plants. Not cross pollination, self pollination. Here we crossed it. Now let us carry out the self pollination. Self pollination of F1 plants. F1 plants are all round yellow. What is the genetic combination? R, capital R, capital Y, small y. Same thing here also. Here these are the gametes here. It is a combination of the shape and the color. Capital R, capital Y, round yellow. Capital R, small y, round and green. Small r, capital Y, small r, small y. The same combination is here. So let us take one plant over here and another plant over here, these two plants. Sometimes we write mother here, father here, or mother this side and father this side. In this case, both parents are of similar type. So the gametes are the same here, ry. The same gametes will repeat on this side also. And you combine these together, you will get the Punnett square, something like this. So in order to understand diagrammatically also in the Punnett square, you can count here round and yellow seeds 1, 2, 3, 4, 5, 6, 7, 8, 9 are there. So round and yellow seeds, plants with round and yellow seeds, you get 9 plants. I hope you understand how to read this capital R, capital Y and all. In my previous dihybrid experiment also we have seen. Only one capital R and one capital Y is enough to make the plant with round seeds and yellow color seeds. So here it is round yellow, round yellow, round yellow. So there are 9 plants for round and yellow. Next you look at this one, 1, 2 and 3 here. These 3 are basically capital R, small y. Capital R for round, small y for green. So 1, 2, 3. There are 3 green plants. So round green, 3 plants. Next comes 1, 2, 3. Small r, capital Y, small r, capital Y, small r, capital Y. Meaning small r represents, the small r represents, what is the small r? Wrinkled. And capital Y represents yellow. So wrinkled yellow, wrinkled yellow, wrinkled yellow. How many plants? 3 plants. So wrinkled yellow plants, 3 here. And finally, this is wrinkled and green, small r, small y is 1 plant. And therefore, if you look at the round green, wrinkled yellow, and if you look at the progeny or the generation, second generation plants over here, all these are the combinations of round, green, wrinkled, yellow. All these independent 4 characters are independently inherited in F2 progeny. So meaning is that in F1 progeny, the small r, small y were recessive, or those got suppressed. Small r refers to wrinkled and small y refers to green. So green color and wrinkled shape were the suppressed in F1 progeny, but they got expressed independently in F2 progeny. So of course, all these traits, individual traits like roundness, greenness, wrinkledness and yellowness got independently inherited in F2 generation plants. So dihybrid experiment is to demonstrate the independent inheritance of the individual traits. I hope you have understood both these experiments. Let us see what is next. So there is one question asked and a topper has answered. How did Mendel explain that it is possible that a trait is inherited but not expressed in an organism? This is a monohybrid experiment, because in F1 progeny of monohybrid experiment, all the plants will be of that kind where the traits are expressed and the recessive traits also will be there. So a monohybrid experiment is the one that explains the traits inherited are expressed in F1 progeny. So what he has written, let us see. Mendel explained this through his monohybrid experiment or cross. He produced progeny from purely dwarf, means purely short, and purely tall plants of P, and he found that all F1 progeny were tall. But when he used F1 progeny to produce F2 progeny, then 25% of the pea plants were short and 3/4 were tall. From this he concluded that both tallness and dwarfness are inherited in F1 progeny, but dwarfness was suppressed under the dominance of the other, which is tallness. Thus he concluded that it is possible that a trait is inherited but it is not expressed in an organism, that is in F1 progeny. Very nicely written, make a note of this. Next, before we understand further the mechanism of inheritance and how do the traits get expressed, we should be aware of certain facts over here. So let us study what are these things. First of all, what are chromosomes? Chromosomes are present in the nucleus, in the nucleolus. Okay, in the nucleoplasm. Nucleolus is the center of the nucleus, and nucleoplasm is the jelly-like substance or a fluid which surrounds the nucleolus. So chromosomes are present in nucleoplasm. So these are long thread-like structures present in the nucleus of the cell, containing the information of hereditary information. What is DNA? See, these chromosomes are made up of DNA molecules. In simple terms, chromosomes are made up of DNA molecules. So deoxyribonucleic acid, it is a chemical molecule in the chromosome, meaning the chromosome is basically a thread-like structure made up of DNA molecules. So, and which carries the information of inheritance. This DNA carries an information of inheritance in the coded form from one generation to another generation. What are enzymes? Enzymes are basically proteins that you should remember is. These are protein molecules in the cells which work as catalyst. What is a catalyst basically? Catalyst is the one which accelerates any chemical reaction without actually taking part into the chemical reaction. So similarly, enzymes are a protein molecules which act like a catalyst inside the cells, and enzymes, these accelerate the chemical reactions, or all biochemical reactions that takes place inside the cell are accelerated or speeded up by enzymes. But the enzymes do not get used up in the process, meaning they are not involved into a chemical reaction. Almost all biochemical reactions in the living things are activated by or carried out in the presence of enzymes. So enzymes are protein molecules, please remember, and these accelerate or speed up the chemical reactions or biochemical reactions in the cell. Gene. In a DNA, it is a long thread-like structure which we call as a chromosome, whereas a small segment of a DNA is called a gene, and gene is responsible for making one protein. So a protein is nothing but enzyme. So can I say here a gene is the one which is involved in making up of an enzyme? Okay, fine. So gene is a functional segment of DNA that provides the information for one protein in a cell, is called a gene. Now, how do traits gets expressed? This paragraph, it's very important. A question can arise. Explain how do traits get expressed. In F1 progeny, the dominant traits got expressed. How is that? That is a question. So let us read this one and try to understand. Cellular DNA is the information source for making proteins in the cell, meaning DNA is responsible for making a protein. A section of the DNA that provides information for one protein is called gene for that protein. So individual protein is governed by one particular gene. That means gene decides what kind of protein is to be made, and the proteins decide the body features. So proteins will control the structure of the body. If there is a variation in the body design, then proteins are different. If proteins are to be different, the gene for that protein should be different. So indirectly we can say the one gene is responsible for making one particular kind of protein. Now, if the proteins, that is the enzymes, work efficiently, the traits get expressed in a better way. Very important. If the enzymes work efficiently, enzymes are nothing but biocatalysts, which are nothing but protein molecules. So if the proteins work efficiently, then the traits get expressed. Keep this in mind. Now, example of tallness. This paragraph is most important in order to explain how do traits get expressed. Example of tallness as a characteristic. Example of tallness is a characteristic. Okay, let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger the growth of the plants. If more hormone is secreted, plant will be taller. If less hormone is secreted, plant will be dwarf. So plant height can thus depend on the amount of particular plant hormone, and the amount of plant hormone made will depend on the efficiency of the process for making it. You got it? If the process is efficient, more hormone. Less efficient, less hormone. So consider now an enzyme that is important for protein making process. Suppose the enzyme works efficiently. If the enzyme works efficiently, a lot of hormone will be made and the plant will be tall. If the gene for a particular protein is such that it has some alterations, and if the enzymes work inefficiently, then less amount of hormone is secreted. That makes the plant shorter, and thus the genes control the characteristics or traits. So read this paragraph couple of times, you will understand. Here in a nutshell, see, height is one of the characteristics. Height depends upon the plant hormone secreted. Plant hormone secreted again depends upon the enzymes basically, or the protein molecules, and a gene controls the enzyme. So if the gene is such that it has got such an information source that the enzymes work efficiently, then more secretions can take place, more hormones can be produced due to biochemical reactions, and the plant grows taller. But if a particular gene is having some modifications, some alterations, etc., so that the protein for a particular protein, the gene of that protein has such an information where the protein will not work efficiently, or a gene for some proteins, or I can say like this, for some enzyme, if the gene has some alteration, then the enzymes may not work efficiently. Why it is so? Because gene has got such an information where it will reduce the efficiency of a particular enzyme. In that case, due to biochemical reaction, less hormone is secreted and a plant becomes a dwarf. So this is how the genes control the traits. Gene controls or contains the information source for enzymes. The gene is such that if the enzyme works efficiently, then more secretion of the hormone, plant taller. The information in the gene is such that it makes the protein or the enzyme work less efficiently, then due to lack of efficiency, hormone secreted is less, plant becomes shorter. So indirectly, gene is responsible for controlling the traits. So this is the answer for this paragraph is taken from the textbook, and make sure you know all these things and make a note of all these points. Next, mechanism of inheritance, very important over here. You must understand over here what is the mechanism of inheritance. In a nutshell, this entire this thing I'd like to revise after reading this one, after going through this one. If both parents help to determine the trait in a progeny, both parents must be contributing a copy of the same gene. If tallness is a trait, then for a tallness, both the parents must contribute a particular type of gene, that is the tallness gene. So if both parents help to determine one particular trait, then both parents must be equally contributing a same copy of the gene. Thus, each pea plant must have two sets of all genes. All the traits of the pea plant are controlled by two pairs of all gene sets. Two sets of all the genes, one set from the mother and one set from the father, or both the sets from two parents basically. So each pea plant must have two sets of all the genes, one inherited from paternal side and maternal side, or from each parent. So each germ cell must have only one gene set. See, remember, the gametes will have, or the germ cells or the sex cells will have half the number of genes present in the parents. So the germ cell must have only one gene set. If there are two gene sets in the pea plants, in the gametes it should have only one gene set. Each gene set is present not as a single DNA but as a separate independent pieces. Each is called a chromosome. That means each gene set is present in the form of chromosome, and every chromosome will have two copies, one from mother and one from father. So that means in the parent you can find two chromosomes, but in the sex cell only one chromosome. One copy of that will be given to the germ cell. So when two germ cells combine, they will restore the number of chromosomes in the progeny, ensuring the stability of DNA. Here, let us see here. I'll take the example of human beings. All the cells of the human beings have 23 pairs of chromosomes. What is this 23 pairs of chromosomes? 46 numbers. What about the sperm and egg? The sperm contains half the number of total chromosomes. In that case, 23 chromosomes. And egg also contains 23 chromosomes. When the sperm fuses with the ovum, 23 plus 23, in a new zygote, 46 chromosomes are restored back. And thus, the stability of the DNA and hence the stability of a particular species is ensured, and therefore the gametes will have half the number of chromosomes as that of in the parents. That you must remember it. So this explains the result of Mendel's experiments and is used by all sexually and asexually reproducing organisms. So this is about the mechanism of inheritance. Okay, make a note of all this. Next, see here the sex determination. That means how the sex is determined in human beings as well as in couple of animals. We'll try to see this. In some animals, like few reptiles, environmental factors such as temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. That means where the environment in which the fertilized eggs are kept, the temperature probably of that environment will decide whether the newborn will be a male or a female. Is it possible for human beings? No. It is possible in case of few reptiles. One more interesting, in snails, individuals can change the sex according to its own will, and therefore indicating that sex is not genetically determined. Very interesting to see here. In the snails, sex determination is not by it is not a genetic process. Genetically it is not determined, but individual snails can change their sex. But with respect to human beings, it is totally different. Let us read this paragraph. This paragraph should be correlated with this diagram. So remember, in an examination, how to explain how to determine the sex, that question will appear. These 4 points along with this diagram is important. Here, instead of drawing the male and female, you can simply say here father and mother and represented by XY and XX. That is enough basically. Or you can say simply parents here, male, female, parents, male, female, gametes and zygote and offsprings. This way you can represent also. What is that? Parents, gametes, zygote, offsprings. These are the sequences which you can represent. So let us read first. What is that? Human beings have 23 pairs, which is 46 numbers of chromosomes in all the cells. 22 paired chromosomes, meaning both the chromosomes are alike, homozygous. 22 paired chromosomes, maternal and paternal copy, one from the mother, one from the mother. So 22 paired chromosomes are there, and one paired is called a sex chromosome, which is not a perfect pair, especially in males. It is not a perfect pair in males. Women have a perfect pair of sex chromosomes, both called X, but men have a mismatched pair in which one chromosome is a normal sized, that is represented by X, and another chromosome which is shorter is represented by Y. So generally women are represented by XX and men are represented by XY because of one paired sex chromosomes which is not a perfect pair. Now, if a sperm carrying X chromosome fuses with an ovum carrying X chromosome, then the zygote formed or the offsprings generated is a female child. So, if a sperm carrying X chromosome fuses with an ovum carrying X chromosomes, then the child born will be a girl. If the sperm carrying Y chromosomes fuses with an ovum carrying X chromosome, then the child born will be a boy. So, let us see here. We know that males are represented by XY because it will have a mismatched pair of chromosomes, especially the sex chromosome basically. So, X is a normal sized and Y is a shorter chromosome, whereas in females it is both XX chromosomes. Now, these are the gametes here. The gametes, either it can have X chromosome or it can have Y chromosome, because only half the numbers will be there. So, it can be either X here or it can be Y here, whereas in case of female, in case of egg, it is only X chromosome. Now, if the sperm containing X chromosome fuses with an egg, it is shown by the combination of X chromosome over here, the zygote formed will lead to a female offspring. So, if a sperm containing X chromosome fuses with egg containing X chromosome, then the offspring produced is girl. If the sperm containing Y chromosome fuses with egg containing X chromosome, then zygote genetic composition is XY. In this case, male child is born. The baby born is male. And this is how the sex is determined. So, please make a note of this and also this diagram. Here, this chapter is complete in all respects up to the portion that has been revised in that has been modified for the current year. That means according to your revised syllabus, up to sex determination is the syllabus that is included. After this one, let me check what is that is. That is, evolution starts from hereafter. But evolution, it is not there. It is deleted from your portions, and hence this is the end of the chapter. In my next lecture, I will be taking up the question answers belonging to this chapter up to sex determination. Okay. Thank you very much for watching and see you in the next lecture.